Our next speaker in session 2 is Chris Fjell. And Chris studied engineering and physics and completed his master's in radiation biochemistry at the BC Cancer Agency, and it was through the Department of Physics at UBC. After several years of working in software development, in industry and academia, he actually went on to receive his PhD in experimental medicine at UBC, and is currently a postdoc in Bob Hancock's lab in microbiology and immunology. He's interested in understanding human disease through a systems biology approach, and he's supported by a CIHR fellowship. Thanks for coming today. Thanks. Help me and welcome you.
I'm going to be presenting this on behalf of Bob Hancock because he's out of town various meetings. So the Pathogenomics of Innate Immunity project, or PI2 project, as you see here, so the project leaders are Bob Hancock and Lauren Babiak. Lauren Babiak is at the Vito Center at University of Saskatchewan. And Jan did the gels component, and Bernadette Mall is the project manager. So I'll start with a few slides just giving an overall perspective of the project and major achievements, and then go into a bit more detail on some of the results. So the main objectives of the PI2 project can be kind of divided into 3 groups. So the first 3 items here are all to do with determining genes, particular genes and their influence on infectious diseases, especially in innate immunity. The major collaborator was the Sanger Institute, who have a large program doing knockout genes in mice. Once knockout mice were generated, the infection phenotype of the mice was determined based on bacterial and viral infection models. We did some work on clinical samples and to look at immune response in humans and other species. And then the second kind of broader objective was to develop novel immunomodulatory anti-infective therapies, as well as a vaccine adjuvant, using a systems biology approach. There was a co-founder for this section, the Gates Foundation Grant Challenges and the FNIH. And the last kind of section is the development of a bioinformatics resource for doing a lot of this work. Integrated into the project was a GELS component to look at the translational genomics of translating academic genomic discoveries into the commercial, civic, and clinical applications that Jan Atkinson Rose Jean worked on with some other people at UBC. So the 3 phases of that were essentially background studies on translational science, survey work, looking at the factors affecting scientists' participation in translational research, and then 3 case studies. The 3 case studies, so commercial, civic, and clinical. The commercial aspect was development of IDR, so that's an innate defense regulated peptide as a therapeutic. I'll describe that in a moment. Civic, open access database in the culture of bioinformatics, and then clinical, translating IRAC deficiency into clinical information. So the objectives of all 3 phases have been achieved, and the findings presented and published. And then just another 3rd summary slide on the major achievements of the PI2 project. So the Sanger Mouse Genomics program generated over 100 embryonic stem cell knockouts, and 33 knockout mice were produced as requested by the PI2 project. And phenotype was determined for several of those. New routes for modulating the innate immunity and inflammatory pathways to do with novel therapeutics and vaccine adjuvants were developed. And the innate defense regulator proteins, there's a word missing there, were studied in several disease systems. Preclinical activities have become on selected IDR peptides, and the first adjuvant has been licensed. The major bioinformatic resource has been completed and is in heavy use by the community, and as I mentioned, the GELS work on translation of genomics is done. Okay, I'll just give a bit of a background on what IDR peptides are. Innate defense regulator peptides are synthetic peptides which are intended to modulate the innate immune system. They're kind of inspired by some of the host defense peptides, also known kind of historically as antimicrobial peptides, that are found in most species of animal. So these are short peptides. The synthetic ones are about a dozen amino acids. The naturally occurring ones are things like defensins, LO37. And for a long time, the host defense peptides were thought of as simply bacteria killers. It turns out that some of them are not, and it's mainly a problem, an artifact of the assays they use to test antimicrobial activity. So they've been renamed host defense peptides. And the innate defense regulators are synthetic peptides that are not intended to kill bacteria. They're intended to modulate the innate immune system to boost the host defense against infection, thereby bypassing some of the problems of antimicrobial drug resistance. So, in terms of the gene knockouts, the milestones were achieved. There were 33 knockouts were requested and generated. The majority have been transferred for work at Vito and UBC. Phenotyping data has been obtained, and with follow-up on the host response to Salmonella, Pseudomonas, and influenza. Future directions, of course, major publication is planned, and then there's a collaborative study with Salmonella and possible continuation of other selected knockout models. So, just to give a sense of what the data looks like, I know you can't read this, but these are the genes across here, and they were assessed wild type versus heterozygous animals, which have been, one gene has been knocked out. And then across here you have various aspects of the animal, fertility, etc., resistance to infection. So Sanger did much of the phenotyping. 33 knockout strains were characterized with respect to typhoid, Citrobacter models. 29 knockout animals or knockout strains in Salmonella, 5 in Pseudomonas model, and 12 for influenza model. So the future directions are more detailed analysis of the selected knockouts and to look at the homozygote cell lines. So moving towards the immunomodulatory peptides, this is an example of the results of in mouse with treatment with 3 peptides. So 1002, 1010, and 1018. These have essentially very little or no direct antimicrobial activity, but they're very protective against Staph aureus in a mouse model. And in a malaria model, the IDR 1018 enhanced survival very significantly. Also protects against TB, E. coli, Salmonella, or MRSA, etc. And a spin-off company has completed phase 1 clinical trials. At the top, this is just a picture of, it's an IVIS picture, so these are luminescent bacteria treated in the animal. And so these are the controls. You can see the large amounts of infection, and then 1018 reduces that to almost nothing. On the vaccine project, the main goal was to come up with an adjuvant formulation which would provide protection against pertussis with a single dose. One of the main problems in vaccinating in developing countries is getting to an area to vaccinate people. So if you can do it in one dose rather than having to schedule somehow to see the same people at specific time points is very advantageous. So just a quick graph showing that the titers were obtained with the triple adjuvant formulation. Okay, so the third kind of broad objective was bioinformatics related. So the main outcome of this was the innate TB resource. So this is a multi-experiment viewer. If you're familiar with the field, Cerebral is a Java plugin for a browser. So what innate TB does, it actually has two main components. It's a resource online for the community to use, which you upload your data. Okay, I'll get to that in a minute. The second one is really a side product of my own work, Metagex, for sort of high throughput meta-analysis of gene expression across experiments. And then data analysis, so to support the other objectives, primarily in ATB has been used with some of the code to Metagex. Challenges for the bioinformatics program is funding. Funding seems to be very hard to come by to maintain software resources. Seems to be a challenge across the field. And also microarray studies and knockouts and continuing efforts in data analysis and manuscript preparation. So an ETB essentially collects all of the public experimentally validated data and puts it in a single resource. As well, a team of curators manually read a large number of papers, so 3,500 papers covering about 3,800 genes, mostly centered on innate immunity, though all of the resources of these other groups were incorporated into innate TB. So just because it's called innate TB doesn't mean that all of the other known cancer information, for example, isn't in there. This little picture here takes some explanation. People tend to be used to looking at signaling pathways as interactions, as you'd see in like a nature review or in the KEGG pathways. From a bioinformatics point of view, if you actually draw the interactions, you end up with a hairball, and that's just how the data actually looks. The hairball for innate DB, for innate immunity, with these other public resources is very sparse. So each dot would represent a gene or gene product, and the lines between them would be interactions. Protein binding or transcription factor binding, that sort of thing. So prior to an ATB curating these papers, this is kind of what it looked like. Afterwards, it's a much more complete picture.